Hello everybody, in today's video we'll be going over your aseptic transfer in order to make your streak plate by using the 4-quadrant method. Making sure that you take the time to perfect your aseptic transfer technique is going to be very important for all of your labs moving forward, because today it might just be a simple lab, but in all of your labs moving forward, you'll probably be asked to make a streak plate in order to get isolated colonies, in order to perform a certain experiment and get a specific result. But it's a relatively simple lab, and as long as you take your time the first time and make sure to pay careful attention moving forward, anybody can master this technique. Starting off on this side, you can see that we have our Bunsen burner, and anytime that you're working with the Bunsen burner, you want to be at your lab desk. Never leave it unattended, and try to bring everything a little bit closer to the Bunsen burner. That way, if anything falls from the ceiling, hopefully the Bunsen burner will burn it and you will reduce the risk of contamination. As soon as your loop turns red, go ahead and let it cool for 10 to 15 seconds. Make sure not to wave it around because that will reduce the risk of contamination. As soon as it's cooled down for that 10 to 15 seconds, you can dip it into the broth, and most likely your bacteria is going to be at the bottom. You're going to see typically some sort of cloudy or hazy material. That is your bacteria. Go ahead and pull it out, and you proceed over to your 4-quadrant technique. Using this coffee cap and a stir as an example, we would take our loop, flame it, wait 10 to 15 seconds, put it into the broth, make sure we grab our bacteria, pull it out, and here we want to keep things as simple as possible. So our streak technique will always be the same, where we start at the top here and then work our way towards the middle. And in order to do that, we will simply turn our dish rather than us going around the dish. Now that we have our microbe here, we will simply crack the top. We don't ever want to open it all the way up. We just simply want to crack the top, give us enough space, and streak. That is going to be represented by this quadrant right here. Then we will turn our plate so that we are now in the 9 o'clock position from the 12. Flame the loop for another 10 to 15 seconds. If you're ever curious to see if your loop is too hot, you can touch it on the inner side of your dish, and if you hear it sizzle or deform a little bit, then you know that it's too hot. If it doesn't do anything, you're good to go for your second streak, which is going into quadrant 1 2 times. So you're starting in there once, go in there twice, and then feather it out, just as we did in this illustration here. Went in once, twice, and then feathered it out. Turn it again, so that we are now in the 6 o'clock position. Flame your loop, wait 10 to 15 seconds. Touch the inside of the dish. If it's good, proceed to quadrant 3, and we're gonna pass through quadrant 2 2 times, just as we did before. That'll be this first lighter green here, where we started in 1, 2, and then feathered it out. Turn to that last 90 degrees. Now the last quadrant, we actually do not need to flame the loop. We can actually, as soon as we turn it, starting in that 3rd quadrant, 1, 2, and then feather it out, as we did over here. So that is gonna be the difference between quadrant 4, is that it is the only quadrant that you do not flame from the subsequent quadrant. The reason why is because there's probably not that much bacteria here anymore, and because you want isolated colonies, there's a chance that the bacteria, there's not that much that will form here, so it's okay if you have a little bit of residual from the previous quadrant. As soon as you're done with that, go ahead and label your plate and store it in the incubator. Go ahead and flame your loop so that it's clean, and then place it away. Let's go ahead and write out those steps. 1 is going to be flame your loop. 2, you'll wait 10 to 15 seconds and then grab your bacteria. Make sure to get it from the bottom, and then you will streak quadrant 1. As soon as you've made your quadrant 1, go ahead and turn the plate 90 degrees and flame your loop again and wait 10 to 15 seconds. So essentially repeat steps 1 and 2. Then you'll make your second quadrant by passing into quadrant 1 2 times. Now repeat steps 5 and 6. So turning the plate 90 degrees, flaming the loop and waiting. Then you will proceed to make your third quadrant where you'll pass through quadrant 2 2 times. 1, 2, and then feather it out. And then 10. You will turn the plate 90 degrees and make your fourth quadrant. Remember, we do not need to flame from quadrant 3 to quadrant 4. But always defer to your lab professor's instructions. As soon as you're done with that, make sure to label your plate, flame the loop so that it's clean, and put away your equipment. Some questions that you want to look out for from this lab are: what is the hottest part of the flame? How long do you leave your loop out for before grabbing your bacteria? Where is the bacteria most likely to be located in your test tube? And then, do you flame your loop from quadrant 3 to quadrant 4? And also, how do you test if your loop is too hot before going into the next quadrant? Those are all questions that are fair game for your lab practicals. If you have any other questions, please leave for me down in the comment section below. And I'm working on some study materials, so make sure to check the description for the links for that. And if this video helped you out, please leave a like and subscribe, and I'll see you guys in the next one.